Hello everyone, my name is Roei Zucker. I'm a graduate student at the Hebrew University of Jerusalem at Michal Lineal's lab, and I'm here to talk to you about finding a sex-specific genetic signal for hypertension disease. Okay, let's start with a bit of background. Hypertension is a disease that is characterized by persistently raised blood pressure. It has a somewhat complex definition that is relied both on systolic and diastolic blood pressure, and it has been called the silent killer because of the myriad of complications it might cause. Some of the complications might be cardiovascular, cerebrovascular, and renal diseases. However, these are not the only ones. As a disease that has to do with blood pressure, many organs that are connected via the bloodstream can be affected by it, and it may cause some lifetime complications. Here we can see a map of the world. What's interesting about it is that there is a very large discrepancy between different continents. Like in the Americas, we can see there is a 23rd moment. In the Americas, we can see there is a 23% of prevalence of hypertension, while in Africa it's 37%. It is considered a very large difference for such a common disease, and it might indicate some genetic background. Another interesting fact about hypertension is the difference between male and female prevalence. For European population, while females have less than 23% of hypertension, males have over 32%. In such a large number, this is also considered very significant. Hypertension itself is considered a multifactorial disease, meaning it has multiple factors that contribute to its prognosis. These factors can either be modifiable factors like obesity, the amount of exercise you do, smoking and nutrition, or they can be non-modifiable like age, ethnicity, genetics, or sex. In this talk, I will focus mostly on the two non-modifiable factors, genetics and sex. And while this disease is highly heritable, the genetic mechanisms behind it are unknown. One reason for why this is unknown could be the effect of biological sex on genetics. Sex-dependent genetics is the way in which your biological sex can affect the way variants or genetics in general are expressed in different phenotypes. It has multiple models in which it can work. It can be either that the variants really have different effect between males and females, or because of a different variant by environment interaction that affects differently males and females. In the most commonly known way, this can happen by the X chromosome. But however, as we said before, some of these ways might not be by the X chromosome, but on any other chromosome. GWAS, as most of us probably know, is a very common tool for associating variants with a phenotype. This tool can also be used to find the sex difference associations. However, GWAS is not without its faults. GWAS only works to associate a variant with a phenotype. It cannot find the interaction between variants, and things like compound heterozygosity will be lost when we use GWAS. For this reason, among others, we decided to use PWAS. PWAS, or Proton Y Association Study, is a tool that was developed in our lab by Nadav Brandeis. It is a gene-based aggregative method, meaning that it aggregates the approximated effect of each variant on protein function to a single score. In this way, each person has a score for each gene, and these scores can be associated with a phenotypic effect. Notice that because the results, the scores are aggregated by genes, we have two main advantages. The first one is that there are a lot less associations we need to perform, which means that we have a much lower statistical threshold we need to stand by, and many of these effects might work together because a gene is usually a single logical unit. Every harm to the gene function or the protein function will probably affect the phenotype in a similar way, which will enhance the genetic signal. Okay, so PWAS's power relies in its aggregation. While every variant has a single score as to how much it might affect protein function, the way these variants are aggregated might differ. PWAS aggregates the variants in two distinct ways, in a dominant or recessive model. In the dominant model, we assume that only a single change is needed to affect a protein function, while in the recessive function, at least two changes are needed, which helps us find the signal that will not be found by GWAS. For this to work, we need to calculate the score of each gene for each person and to associate it with a phenotype. To do so, we need to use a dataset that contains both these properties. For this, we use the UK-Biobank. The UK-Biobank is a very large a biobank that contains both genetic, medical, and lifestyle information about its participants. It has over 500,000 participants, which in the age of recruitment were aged between 40 and 69. The age of recruitment was the time of recruitment was about 15 years ago, meaning that most of the people that will develop hypertension probably developed it by now. We filtered out some of the participants to avoid biases. We only used participants of European descent, and we did not include participants whose biological sex did not agree with their reported sex. Also, we chose only a single participant of each family group to avoid further biases. After this filtration, we had 125,000 males and 150 females, either case or non-cases. And as for the phenotypic measurements, we've used a diagnosis of primary hypertension and both diastolic and systolic blood pressure measurements. After the filtration, we've also had 40,000 males and 33,000 female cases. Okay, so as for results, here we can see the PWAS analysis. Remember that we work here on the protein level, meaning that each of the number here represents a gene that was found significant. We can see here a Venn diagram of each sex group that we included, either running the analysis only on males, only on females, or on both males and females. What's really interesting here to see is that females had a much stronger genetic signal. While there are more male cases, there were only 2 genes that were found significant for males, and over 20 that were found for females. Most of the genes for females and all of the genes for males were found with me when we performed the analysis both on males and females. However, there were 4 genes that were only found significant in females. This indicates there might be some specific genetic signal that is unique for females. On this diagram, we can see a p-value comparison, the minus log of p-value between males and females, and we can see here the same pattern remains. The genes that were found significant in either males and females, most of them seem to be clustered near the female significant part, meaning that the results are much more significant for females. In these 2 bottom figures, we performed the same analysis, but instead of using the primary hypertension medical diagnosis, we use the systolic and diastolic blood pressure measurements. We can see that the same trend persists, a very much stronger female signal, and we can see that there is unique female genes that are found only when we perform the analysis on females. Notice that while systolic and diastolic blood pressure are very closely tied with the diagnosis of hypertension, they are not exactly the same, which makes it a somewhat good validation. Here we try to look at what might be the reason, what might be the underlying story that causes females to have such a different genetic signal. We've looked at the top 10 most significant genetic top 10 most significant genes that were found for females, and we found that all of the genes have some sort of relationship with either systems or tissues that have to do with the immune system. We can see here that almost all of them are either very much expressed in tissues that have to do with the immune system or have a known immune function. We also wanted to validate our results using independent sources. We've used different data sets that performed similar analyses trying to find the genes that have to do with primary hypertension or other blood pressure related values. And we found that over almost 30% of the genes that we found were also found in other sources, and that's 22 genes. This number is very encouraging for us because such numbers are not usually found in replication. In this graph, we can see how much intersection there was between the different sources, and we can see that in many of the cases, these genes were found more than once, so the results are very reliable. We also wanted to perform validation of our results using coding GWAS. GWAS, as we know, is not a gene-based method, it's a variant-based method. So what we did, we performed GWAS only on the coding region of the genes, and if a variant was found inside a gene to be significant, we've concluded that the entire gene is found significant. Here we can see the comparison we did with the PWAS results we've got, and more than half, almost 2/3 of the results, over 2/3 of the results were found to be significant, 16 genes intersection with PWAS. We've also performed the same analysis we did on PWAS. We looked on the different sex groups and tried to find if the trend persists, which it did. Most of the signal, it comes from females, a small signal from males, and a female-specific unique signal. However, because the results are much smaller, we can't base anything significant on it. Finally, we performed enrichment analysis. In enrichment analysis, we tried to find if there are any tissues in which these genes are more expressed in a significant way. What we found is that in almost every case, the topmost tissues that were found to be enriched were endothelial tissues that have to do with the vascular system. This is very encouraging to us, and it really shows that the results indeed have to do with hypertension. What's really interesting is that for diastolic blood pressure in females, we found that a macrophage spleen was found to be significant. It's very interesting because it has very much to do with the immune system. So it really strengthens the idea that the female-specific signal has to do with the immune system. Okay, so in conclusion, hypertension is a multifactorial disease, and it has a somewhat stronger genetic signal. But this genetic signal is different between males and females. Much of the genetic signal that is found in females is very associated with the immune system. And this indicates there might be a need for different diagnosis or treatment for males and females. There were some limitations, however, to our study. There are multiple sex-dependent models that exist. It could be that really the genes are only relevant in females or only relevant in males. However, it might be that males and females are exposed to different environments, in which way maybe the effect of the genes is the same in males and females, but a different environment causes the difference. And in our study, we've also only used the common imputed variants from the UK-Biobank. We didn't use rare variants which hold a lot of genetic power, explanatory power. And we've also only used coding regions when we know that the intergenic and the non-coding regions have a lot of regulatory effect on phenotypes. Finally, we've only used participants of European descent, which hypertension is not only a European issue. Running the same analysis on non-Europeans might show us some very interesting results. Future directions in general, we want to create a method that will help us find other sex-related phenotypes other than hypertension. All of these analyses can be applied to other phenotypes. We just need to do it. We also want to perform the analysis using exome sequencing. The UK-Biobank has it for almost all of its participants, and it will be interesting to use it. And finally, we have the PWAS Hub. It's a website developed by our lab to show the PWAS results of these analyses and many other diseases. It will be online shortly. And there is a paper published on this study. So thank you. I'd like to thank you very much for listening. I'd also like to thank the members of my lab, especially my colleague Miki and my PI, Professor Michal Inyal. And I'm open to any questions you might have.
Thank you so much. Thank you for keeping the time, even though you had to restart. So we have time for 1 or 2 questions. If there are any questions, please come down here. So I also had a question on your last slide. It was really nice that you highlighted, maybe it was the second to last slide, that you highlighted the limitations. I'm wondering if differences in sample size could also play a part.
Yeah, the reason we use the European descent is because the UK-Biobank has mostly European participants. We would really like to apply these results to other ethnicities. We also know that there are some treatments that are unique for the Afro-American, for Afro-Americans, for hypertension. So there is very likely there is a different genetic signal. However, the number of participants is so small, we couldn't perform the same type of analysis.
All right.